The overall goal of the following experiment is to evaluate the effectiveness of cancer drug sensitization by measuring changes in cell metabolism in vitro and metastatic frequency in vivo. This is achieved by first transfecting cancer cells in vitro with antisense molecules to knock down the target of interest. Next, the transfected cells are plated onto biosensor chips and loaded into a metabolism measurement system to enumerate changes in acidification, respiration, and adhesion during drug treatment. Separately, transfected cells are treated with a drug and injected into the venous circulation of a chicken embryo in order to evaluate effects on metastatic frequency. The results show cancer drug sensitization as illustrated by changes in cell metabolism and metastatic frequency.
This technique extends the development of cancer therapy because it allows for the simultaneous evaluation of parameters that are not always considered during conventional preclinical testing of drug targets or compounds.
Though this method can provide insight into novel drug targets involved in DNA repair, it can also be applied to target validation in other systems, such as cell cycle control.
After transfecting tumor cells with antisense oligonucleotides according to the text protocol, in a sterile environment, use 400 microliters of PBS to carefully wash 6 biosensor chips. To sterilize the chips, remove the PBS and add 400 microliters of 70% ethanol. Incubate for 20 minutes before using PBS.